Hello everyone, today we have a very important lecture and it's in the series of Let's Read the Anatomy for Dental Students. The subject is folding of the embryonic plate and its sequences. As already indicated, the endoderm forms the cells that line the respiratory and gastrointestinal tracts. The flat embryonic disc has to become folded if the endoderm is going to be a suitable location to become these linings. In earlier stages of development, the embryo is a flat plate of cells differentiating into the different germ layers. The growth of ectoderm during formation of the neural tube and of the mesoderm during segmentation has been described. This growth has profound consequences for the relative positions of the embryonic germ layers. Profound consequences for the relative positions of the embryonic germ layer and specific structures formed from them. Most of the growth takes place on the dorsal surface initially, and the principal effect is that the embryo bulges upward into the amniotic cavity. The resulting folding or flexion of the embryo is not confined to the transverse axis but occurs longitudinally to it. We will follow a series of diagrams illustrating the folding of the embryo in longitudinal and transverse sections. To observe consequences of folding on the relative position of different organs and tissues. Longitudinal folding, a series of sequential sagittal sections of the embryo starting with the flat embryonic disc. The following features can be seen in sequence. Moving from left to right on the diagram, which is from head to tail on the embryo. Converging layers of ectoderm and endoderm. A region of lateral plate mesoderm between the ectoderm and endoderm known as septum transversum. The primitive pericardial cavity and the intramembranic column. The buccopharyngeal membrane. lateral palate mesoderm in future septum transversum. Neural plate, notochord, primitive pericardial cavity, ectoderm, endoderm, yolk sac, longitudinal folding of the embryo, buccopharyngeal membrane, cloacal membrane, midgut, foregut, hindgut, developing heart, buccopharyngeal membrane ruptured, vitelline duct, vitelline duct. A sandwich with neural plate above, notochord between and Indoderm below, forming most of the length of the embryo. The cloacal membrane, deriving layers of ectoderm and endoderm. You can also see as you study the sequence of diagrams comprising that the neural plate expands considerably at the head end to produce the rudimentary of brain, displacing the pericardial cavity inferiorly into the chest region as the neural plate expands anteriorly and posteriorly to form the central nervous system. The longitudinal curvature of the embryo increases and the cavity position of the other tissue changes. As shown, the buccopharyngeal and cloacal membranes become folded under the superior and inferior ends of the embryo, respectively, and the process the reto so that their originally ventral surfaces come to face dorsally. As the curvature continues to increase, part of the yolk sac becomes incorporated into the embryo to become the gestational tract. The communication between the gut and yolk sac becomes constricted as the folding becomes more pronounced until it eventually reduced to the narrow vitelline duct, which will form the umbilical cord. Note also the radical alteration to the position of the pericardial cavity as folding takes place. It moves from its position anterior to the embryo shown to lie beneath the superior part of the gut, foregut. In the future chest region shown, the septum transversum also moves to lie between the pericardium cavity and the enclosed yolk sac. Eventually, the diaphragm and the liver will form in the septum transversum, the buccopharyngeal and coical membranes. The only areas where the buccopharyngeal, cloacal membrane, neural groove, ectoderm, somite, amniotic cavity, colon, heart tube, Indoderm, yolk sac, neural tube, aorta, intra-embryonic colon, amnion, vitelline duct, yolk sac, aorta, peritoneal cavity, foregut, colon, liver. Ectoderm and endoderm are not separated by the mesoderm, come to lie at the beginning and end of the forming gastrointestinal tract. You can see that both membranes break down so that the gastrointestinal tract is now open at both ends. The superior end is the position of the future oral cavity and the inferior end is the position of the anus. Transverse folding. A series of transverse sections through the flat embryonic disc are shown. Identify the amniotic cavity, then the following layers, starting from the base of the cavity and progressing downward. The ectoderm thickened centrally to form the neural plate. The notochord centrally flanked by blocks of mesoderm, somites. Note how the lateral plate membrane splits laterally to form two layers, continuous with the extra-embryonic mesoderm, separated by the intra-embryonic colon. The endoderm, the greatest growth, is the neural plate. By comparing, you can see that increases in size of the neural plate causes the lateral margins of the embryonic disc to rotate downwards about an axis approximately through the notochord. This movement incorporates part of the yolk sac into the embryo as the gastrointestinal tract, as already seen during the longitudinal folding. The lateral margins of the folding disc eventually come into contact as transverse folding proceeds. The part of the yolk sac enclosed by the lateral folding of the embryo forms a tube to become the gastrointestinal tract. The intraembryonic colon will form the peritoneal cavity in which the gastrointestinal tract and its associated organs will develop. The ectoderm also grows around to form the outer covering of the entire embryo. This is a brief overview of the complex changes that occur to generate a whole organism from a single cell, the formation of the embryonic germ, layers, and their derivatives are summarized diagrammatically. Figure gives examples of the ways in which diseases and therapeutic drugs can affect the complex sequence of events in the early stages. The detail of how specific tissue and organs are formed by the interaction of the embryonic germ layers will be described in the context of their anatomy. Keratogenic effects of Drugs and disease. As we have seen, the complex processes of development require precise coordinating. It can take very little to disturb these processes and the results can be serious. The causes of many developmental defects are unknown, but the causative agents of some conditions have been identified. Such agents' defects are called teratogens, from the Greek monster forming. Two examples are given, although they actually affect events outside the first 3 weeks. They are graphic illustrations of keratogenics, many of which probably act in the time window of this chapter. Morning sickness often affiliates pregnant women and is usually most prevalent in the 6th to 5th weeks of pregnancy. The limbs are developing in the early part of this time. In the late 1960s, a drug, thalidomide, was developed to relieve the symptoms of morning sickness. The drug proved to have disastrous side effects and interfered with the processes that control the formation of the limbs. Many babies born where thalidomide was prescribed were born with malformations of arms and legs. As a result, the females in developed countries are now immunized against rubella, germ, and measles. At around the time of puberty, germ and measles in itself tends only to produce a rash and high fever, and the general feeling of being unwell and lasts at most only a week or two. If pregnant female contracts rubella up to 12th week of pregnancy, infection by the rubella virus may produce congenital rubella syndrome. There are 3 common malformations which may occur singly or in combination. The inner ear is affected in about 60% of cases, resulting in hearing loss. The eyes may be small or even cataracts, 30%. Heart development, 50%. Patient duct arterioles is a frequent manifestation of heart malformation. Folding of the embryonic plate and its consequences. Epiblast, ectoderm, intraembryonic mesoderm, Indoderm, neuroectoderm neural tube, paraxial, intramediate, lateral. Ectoneuro, ectomesenchyme, neural crests. Whole axis, head and neck only, trunk only, sensory nerves and ganglia, autonomic nerves and ganglia, Schwann cells, melanocytes, skeleton meninges, muscle connection tube, adrenal medulla. Ecto, epidermis, epidermal appendages. Emberionic mesoderm, intermediate lateral paraxia. Dermatome, sclerotome, myotome, cardiogenic mesoderm, dermis, bone, cartilage, skeletal muscle, circulatory system. Intermediate, urinary system, reproductive system, lateral, primitive, circular system. Endoderm, epithelial lining of respiratory GI tract. The origins of embryonic germs, layers of their derivatives. So this concludes this page and this lecture and this section. Next section of the book is the thorax. Thank you for listening and goodbye.